Hi everybody, in this lecture we study about proteins. Proteins is the building blocks of our body, and at the same time it's the functional materials such as the enzymes. For this lecture also I selected some references from Wikipedia. In this lecture we are going to learn the basic structure of proteins and the relationship of protein structure with information gene code. And the synthesis of the protein we call translation, the main players of that are the translation and also how they work. After that, we are going to study the details chemistry, what's going on in the middle of protein synthesis. And then we are going to move to proteomics and post-translational modifications. Now let's move into protein structure. This is a central dogma of modern molecular biology. We believe those genetic information is stored in DNA, and the information, some part of it, is transcribed into RNA, and then eventually that RNA information is translated into protein. So we define that one information to make one protein that is defined as a gene, and that is stored here, and one unit of that gene is transcribed into RNA, so in terms of mRNA, right? After that, that information is eventually expressed in the form of a protein. So for each stage, if DNA make exactly same copy of that the structure, that process we call replication. So there is a DNA replication, and then the enzyme responsible for that we call DNA polymerases. For the transcription, we need to use RNA polymerase. So that is using DNA as a template and make RNA. Eventually, using this RNA information, the machinery which make a protein is ribosome, and this translation is a process of using RNA information to make a protein. So this is kind of a central theme, central principle of the biology. That's why we call this as central dogma of molecular biology. In addition to the basic central dogma, we found several exceptions. So for example, there is some viruses, they use RNA as their genomic information. Sometimes that viral the sequence is the same as gene, then we call this the RNA as positive sense the RNA. But sometimes the genome of the virus is complementary sequence of this the sense RNA, then we call this as a negative sense RNAs. So in this case, this should be template of that the gene expression. Now if this RNA, if they replicate, they can make DNA from RNA. The original central dogma says only RNA can be made from DNA, but this is actually this is the opposite process, utilizing RNA as a template and then synthesize that DNA. So this is kind of a transcription, but actually this is the opposite way. That's why we call this other enzyme as a reverse transcriptase and this process as a reverse transcription. Even later, we found that it's also possible to synthesize RNA copy based on this the RNA template. And in this case, this is a RNA templated RNA synthesis. In a sense, we can call this as RNA replication. That's also possible. In either cases, once we make this DNA from the RNA template, and then DNA can form double helical structure, and then eventually they make more copies of this RNA strand. So this kind of exception was found in addition to the basic central dogma, and has been utilized for many the interesting biological studies. For RNA synthesis, some activating factors should bind to this promoter region and then start those RNA synthesis from here. After RNA is synthesized, the intron should be removed, and then on the 3' area, poly A tail is added. Then matured mRNA can be synthesized. After the amino acids are polymerized, so-called protein is synthesized, and proper folding and also post-translational modification happen to make functionally active proteins. The building blocks of a protein is amino acid, and this is a basic structure of amino acid and should be very familiar to you, right? So amino acid is composed of one amino group attached to the central carbon and then one motif of acid. And that's why this is called as amino acid. Depending on the position of attaching of this amino group, it can be called as alpha or beta. So in this case, if this is attached directly to the central carbon, we call this amino acid as alpha amino acid. When amino acid conjugated with another amino acid, the reaction happens between one carboxylate and one amine groups. And they react together, and by removing one molecule of water, and then they form this peptide bond. Peptide bond is composed of one CO group and NH group connected together. I'm sure this is very basic knowledge for you, right? Still, you should be very familiar with this structure. And how many amino acids are in this peptide? Can you figure out? Yes, there are 3 amino acids conjugated together, so you can call this as a tripeptide. So here, this is a 1 amino acid here, and second amino acid, and third amino acid, and then this bond connected together, CONH group, this is the so-called the peptide bond. Now these proteins, we can give direction. So amino side, we call this end as a N-terminal, and then the other side of a carboxylate side, this should be called as a C-terminal. So for any of those protein structure, this is a convention starting from the N-terminal to C-terminal directions. This is a structure of 20 amino acids found in our proteins. So if you look at the structure here, this is amino acid, and then you can look at this part, this is side chain. And alanine case, this is a methyl group. Glycine case, this is a proton. So that's why this molecule is not chiral. And this is smallest amino acid we have. And then if this chain they're getting longer, this is leucine. And then if this methyl group is moved to this direction, then this is the isomer to each other, but this is a different position, so we call this as isoleucine. So interestingly, isoleucine has 1 and 2 chiral centers. And many of them is just the simple primary alkyl group. Actually, the proline case, this alkyl group is connected to amine. So other amino acids are all primary amine, but proline is a secondary amine. Interesting, isn't it? And valine is also another alkyl, other chain, other amino acids. So this amino acids is considered as aliphatic other amino acid. Some other amino acids, they have aromatic groups like phenylalanine and tryptophan and tyrosine like this. And others have this carboxylate. So this is acidic other amino acid. So aspartic acid and glutamic other acid. Interestingly, also we have this amino acid is formed amide bond. So this is asparagine and glutamine. It's the derivatives of this these 2 acidic amino acids. As a basic amino acid, we have arginine, which has a guanine motif here, and histidine, and also we have lysine. And 2 amino acids contains hydroxyl groups, and 2 amino acids contains sulfur groups. So this is general structure of amino acid, and depending on those the functional groups, it can be categorized with a different color. And many of them, about half of them, can be synthesized in our body, but the other half of them, we cannot synthesize them in the body. That's why we should take them from food. This kind of the amino acid we call essential amino acid. Let's just think about the information system. Our computer system is composed of a binary system. That means all the information is expressed either as a 1 or a 0. If you use 2 units of this information, we call this system as a 2-bit system. If it is a 2-bit, how many different information we can store in this 2-bit unit? It can be 00, 10, 01, or 11. So that's why in this case, this is a 2 by 2. We can store 4 different kinds of informations. If it's a 4-bit, connecting 4 together, how many information we can store here? That one is a 2 by 2 by 2 by 2. It can be 16. How about the 8-bit? Then it can be 16 by 16. That's 256. If it goes further, let's say 16-bit, now it can be quite big. It's bigger than 65,000 different combinations. So our current computer systems, usually we use this 16-bit as a basic unit of the information, the coding. So this can code all different alphabets and also numbers kind of things. But more and more, the computer system becomes more complicated. Now you may have heard 32-bit or 64-bit, kind of a bigger unit to express different informations. Now, if you wanted to utilize this binary system to code 20 different amino acids, then the question is, what is the minimum bit size for encoding 20 amino acids? If you think about this 4-bit, now this one has only 16. So we have a 20 amino acids, and this is not enough number, right? Then how about the 5-bit? Yeah, if it's a 5-bit, then maybe we can encode 32 different information. Then maybe that's enough to code 20 different amino acids. Then, if 20 has been used, still we have the 12 left over, right? How can you use those 12 leftovers? There may be different ways. You can use them, maybe encoding one amino acid with multiple choices of informations. But let's think again. If this is your binary the code system, it looks like matrix the information, right? Then how do you know which one is a starting point and which is an ending point? That means where do you put your space in between of those one information unit? So computer system also has exactly the same problem. That's why usually they have some designated the unit. Let's say all 0. Maybe this can be used as kind of a starting point. And maybe all the 1, then maybe this can be used as maybe ending point. Then you need to assign at least 2 units, one for starting, one for ending. Then out of 32, you used up 2 and 24 amino acids. Then you have only 10 leftovers. If you think about the biological system, our biological system, we utilize not binary system. Actually, we have 4 code. In RNA case, we have A, U, C, G, 4 code. So we can call this as quaternary system. Then the question is, how many different nucleotide is required to encode 20 amino acids? In DNA RNA case, also we have the direction, right? So we can call those directions from 5' to 3'. So you cannot read the opposite directions. Now the question is, what is the minimum size of a bit to encode these 20 amino acids? If it is only 1 bit, you have only 4 information units. So it's not enough, right? How about the 2-bit? 2-bit, you have a 16. Is that enough? Actually not, right? Then how about the 3-bit? Yes, now you have more than 20, 64. Then even though you utilize the 24 amino acids and maybe 2 for 1 starting, 1 ending, still you have enough numbers of information to cover to encode the old amino acids. So actually, that's correct. Our biological system is also utilizing minimum 3-bit system here. 3-bit as encoding system for one amino acid. So in the sense, we can call our biological machinery system is 3-bit computer. Interesting, isn't it? We utilize a little more complicated quaternary system, not binary system, but information the size-wise is shorter, so 3 nucleotide can define one amino acid. So we have the direction of this nucleotide from 5' to 3 at the 3' end. So same directions we give here. So we call starting point here is N-terminal and the ending point as C-terminal. This is how we give those orders and directions. Now next question is, how did we know all of these possible 64 combinations? Which combination, which 3-bit of the information is designating which amino acid? That's a good question, right? These two scientists, they solved the problem. They started with a very smart experiment like this. First of all, they made polymers with only one nucleotide, U. Then it doesn't matter where is the starting point. Anyway, those information should be composed as same unit of 3 other bit information, U. And then they expressed the protein utilizing this RNA sequence, and they found they could get polymer of phenylalanine, Fe. So that means clearly this shows that U is designating phenylalanine. So this is the starting point of their work. This historic experiment has been done in 1961. And at the moment, the supervisor was Dr. Nirenberg, and he was working in NIH. And this is their lab notebook. Currently, this is displayed at main entrance of NIH at the campus. And utilizing this experiment and expanding this, and they decoded all the genetic information, which gene is making which proteins. So, of course, this historic experiment that resulted in the Nobel Prize awarded in 1968. So utilizing this information, now we can say the genetic code which is designating for each amino acid. Now here you said U, right? So U case we can find starting from here U. This is the phenylalanine. So similar way in here UUC is also designating phenylalanine. Now if you change it UUA, UUG, this is the leucine. So in this system, now we can make all code for different other amino acids. Now you can see here, out of those 64 different other combinations, actually multiple informations is coding one amino acid each. Now this is a summary of amino acid and also it's a genetic information code. Let's just say alanine case, and it has GCU, GCC, GCA, GCG. So at least the first two is similar, right? First two is the same, and then third one can vary a little. Now this is the genetic code for alanine. Easily we can see here, some amino acid has more genetic coding information than the other. That means we can assume that maybe alanine has been used the more than the other. Like cysteine has only 2 code and alanine has a 4 code, and we can correlate that occurrence percentage of that amino acid in the protein. In human case, let's just say alanine is about 8%. Cysteine is only 2%. That makes sense, right? So the biggest one here, let's just say this one, leucine as a 6, it's about the 9%, and here arginine is 5%. That's interesting. So even though those genetic code number is the same, 6 and 6, sometimes the occurrence of the amino acid is not exactly matching. One of those most interesting other amino acids for the chemical biology is cysteine and the lysine, because they are nucleophilic amino acid. So if you look at here, cysteine is only 2% with the 2 genetic coding the information. Lysine also has only 2 information, but it has almost a triple, 6%. So this is worth memorizing. Lysine has about a 6% occurrence. Cysteine has about 2%, one of those lowest, next to tryptophan. Okay. Now here we can find also stop codon. 3 of those sequences has been assigned to the stop site. And you may ask, what is the starting point, right? So starting point, the interestingly, is overlapped with this methionine sequencing, so with the AUG. So with the AUG, we start the protein synthesis. And most of those protein that are synthesized, they start with the methionine. Interesting, isn't it? I said before, we have 20 basic amino acids in the protein, but this table is from recent Wikipedia. Can you count the amino acid here number? Actually, it's more than 20. It has a 22. Which one is non-classical amino acid? So by the way, each amino acid, it has a name: alanine, cysteine, aspartic acid, and then it has a code as three character abbreviation: ala, cis, asp, glue, kind of a 3 word, and also it has a 1 word, the decoding the symbol, encoding symbol. So this is the worth the memorizing to accelerate your understanding of the reading protein structures. Can you find extra 2 abnormal amino acid here? Did you find it? Yeah, one is here, pyrrolysine. The other one is selenocysteine. These 2 new other amino acids from some species, not every species, they utilize some of those stop codon, not as a stop codon, they utilize this as coding information for this strange abnormal amino acids. So now we should think whether we should include these amino acids is also the basic amino acids in our protein structures. So that's why sometimes we call this the 21st or 22nd amino acid in the protein system. So knowledge system is becoming more and more complicated and enriched. So far, still, usually we say we have 20 amino acids in the system, but maybe that knowledge can be changed. Even our textbook may be changed in the future. Here is another summary of our 20 amino acid structure. Now let's look at it again. Glycine and alanine is kind of considered as a small amino acids, so which has 1 proton, and this one has the methyl group. And nucleophilic other amino acids, we have hydroxyl group containing serine and threonine, and cysteine has the thiol group. And also we have a hydrophobic amino acids like valine, leucine, isoleucine, or methionine, or proline. And also we have 3 aromatic amino acids and 2 acidic amino acids and 3 basic amino acids and 2 amide containing amino acids. It's amazing to see, utilizing only these 20 amino acids, we build up all the protein structures, most of those protein structures, and then make out so many different functional other proteins, only utilizing these 20 the building blocks. It's amazing, isn't it?